Assalamu alaikum, friends. Today I'm going to start a series of lectures on lipid metabolism, and this is my first topic and first discussion on lipid metabolism. So, there are some keywords related to the digestion and absorption of lipid: emulsification, bile salts, pancreatic enzyme, mixed micelle formation, absorption through simple diffusion, and Chylomicron. Okay, so dear students, वन बाय वन हम इनको सेपरेटली डिटेल से डिस्कस करेंगे, लेकिन फिलहाल इनके हम मीनिंग्स को अंडरस्टैंड कर लेते हैं. इमल्सिफिकेशन, मस्ट रिमेंबर, मैं अगेन रिपीट कर रहा हूँ, मस्ट रिमेंबर दीज कीवर्ड्स. Emulsification means the dispersion of larger lipid molecule into simplest form. Emulsification means the dispersion of larger lipid molecules into simplest form. But there are some requirements for the emulsification process. There are some requirements like bile salt, amphipathic lipid, and peristaltic mixing in intestine. Emulsification means dispersion of large lipid molecule into simplest form, and there are some requirements for emulsification process: biosalts, amphipathic lipid, and peristaltic mixing in the intestine. Okay, dear students, and this is just the little bit meaning of emulsification. Next, bile salt. Now, bile salts. Bile salts, they are just sodium and potassium salts of tarocolate, tarochinodioxycolate, glycocolate, and glycochinodioxycolate. Bile salts, they are just sodium and potassium salts of glycocolate, tarocolate, glycochinodioxycolate, tarochinodioxycolate. Bile salts, they are just sodium and potassium salts of glycocolate, tarocolate, glycogenodeoxycolate, and tarogenodeoxycolate. Cholic acid and chenodeoxycholic acid, they are bile acids. Conjugation occurs with the help of glycine and taurine, and they are converted into glycocolate, tarocolate, glycogenodeoxycolate, and tarogenodeoxycolate. Now, bile salts, it is very important. It reduces surface tension of lipid. It reduces the surface tension of lipid. Listen, this is the suppose this is a lipid molecule, and after this smallest lipid molecule, and here is the role of bile salts, and this increases the surface area of lipid molecule, and here enzyme can easily act. Okay, and also in mixed micelle formation, it plays a very important role. Bile salts, they are just sodium and potassium salts of glycocolate, tarocolate, glycogenodeoxycolate, tarogenodeoxycolate. It reduces the surface tension of lipid, and in this way enzyme can easily act on this lipid molecule by due to increase in the surface area of lipid molecule, and it is very important in mixed micelle formation. Next is pancreatic enzyme. Pancreatic enzyme, they are very important. Pancreatic enzyme, for example, there is release of some enzyme from pancreas like cholesterol esterase, phospholipase, and lipoprotein lipase. These enzymes are secreted from pancreas. Cholesterol ester enzyme, it acts on cholesterol ester. Phospholipase enzyme, it acts on phospholipid, and lipoprotein lipase enzyme, it acts on lipoprotein. Okay, dear students, the enzyme and their function. Next is mixed micelle formation. Mixed micelle formation. Mixed micelle mean combination of digested product and bile salts. Combination of digested products and bile salt, it is called as mixed micelle, and all these products, they are absorbed by simple diffusion. Okay, and they gets incorporated in chylomicron formation. डियर स्टूडेंट्स, अब इनको हम डिटेल्स डिस्कस करते हैं किस तरह से डाइजेशन का प्रोसेस होता है और किस तरह से एब्जॉर्प्शन का प्रोसेस होता है. ओके, लेट्स स्टार्ट. Digestion. There are basically three sites for the digestion of lipid. The first one is mouth. Mouth, there is negligible digestion of lipid. Why? माउथ में किसी किस्म की डाइजेशन लिपिड की नहीं होती, क्योंकि जो हमारे टंग की डोर्सल सरफेस से जो एंजाइम रिलीज होता है लिंगुअल लाइपेज एंजाइम, उसको इतना टाइम नहीं मिलता कि वो लिपिड के मॉलिक्यूल पर एक्ट कर सके। इसी वजह से ओके, ड्यू टू लेस टाइम, क्योंकि जो ट्रांजिट टाइम होता है एंजाइम की वर्किंग के लिए वो बहुत ही कम होता है, इस वजह से माउथ में किसी किस्म की डाइजेशन नहीं होती। नेक्स्ट इज स्टोमक। स्टोमक में भी देखें दो तरह के एंजाइम होते हैं, लिंगुअल लाइपेज लाइपेज एंड गैस्ट्रिक लाइपेज। Both enzymes they work only on short chain fatty acid and medium chain fatty acid, and such type of enzyme are present in infants and young children. Okay friends, they do there is not any kind of enzyme in stomach that act on triacylglycerol due to long chain of hydrocarbon. In stomach, lingual lipase, gastric lipase, they are active only in infants and young children. They act only upon short chain fatty acid and medium chain fatty acid having 12 carbon atom. They do not act on the triacylglycerol molecule. Next, intestine. Intestine. It is a main site for the digestion of lipid. Main site. The lipid digestion starts from here. When chyme reaches to intestine, then there is a release of two important hormones, cholecystokinin and secretin. Cholecystokinin and secretin. Cholecystokinin, it is responsible for the contraction of gallbladder and hence responsible for the release of bile. And it is also very important, it acts on pancreas and responsible for the release of pancreatic enzyme, cholesterol esterase, phospholipase, and lipoprotein lipase. Secretin, it is responsible for release of bicarbonate ions and it neutralizes the acidic pH of stomach. इंटेस्टाइन में अल्कलाइन कंडीशन होती है, पीएच तकरीबन 9 पीएच रहती है, 9.5। मैं क्या बात की रिपीट करता हूँ, इंटेस्टाइन में कंप्लीटली डाइजेशन का प्रोसेस होता है और यहाँ पर दो इंपॉर्टेंट हार्मोन रिलीज होते हैं, cholecystokinin and secretin. Cholecystokinin, it is responsible for the contraction of gallbladder and release of bile, and it acts called pancreas and responsible for release of pancreatic enzyme, cholesterol esterase, phospholipase, and lipoprotein lipase. Secretin, it is responsible for release of bicarbonate ions and hence it neutralizes the acidic pH of stomach. Now, next, reactions. Listen. This is acyl CoA and this is glycerol molecule. And collectively they form the structure of triacyl glycerol. What happens? Here important enzyme acts and its name is lipoprotein lipase. This enzyme acts upon carbon number 1 and carbon number 3 of triacylglycerol molecule and converts it to triacylglycerol into two molecules of monoacylglycerol and two molecules of free fatty acid. Okay, dear friends, triacylglycerol molecule acted upon by lipoprotein lipase. It acts on carbon number 1 and carbon number 3 and converts it into two molecules of monoacylglycerol and two molecules of free fatty acid. Okay, next is phospho. There is action of an important enzyme phospholipase and it acts upon carbon number 2 of this phospholipid and converts it into lysophospholipid and free fatty acid molecule. Phospholipid acted upon by lipoprotein acted upon by phospholipase enzyme that acts on carbon number 2 and converts it into lysophospholipid and free fatty acid. Third one is, this is first one, second one, and third one is cholesterol ester. It is acted upon by cholesterol esterase enzyme and converts it into cholesterol and free fatty acid molecule. तो ये है डाइजेशन का प्रोसेस. अब यहां से डाइजेस्टेड प्रोडक्ट हमें ये मिल रहे हैं, two molecules of monoacylglycerol, free fatty acid molecule, lysophospholipid and cholesterol. ये हमें डाइजेस्टेड प्रोडक्ट मिल रहे हैं. कहां पर डाइजेशन हुई है? इनकी इंटेस्टाइन में हुई है. दो तरह के हार्मोन बहुत इंपॉर्टेंट रहे हैं, कोलिसिस्टोकाइनिन एंड सेक्रेटिन. कोलिसिस्टोकाइनिन ने क्या किया? कंट्रैक्शन ऑफ गॉल ब्लैडर एंड रिलीज ऑफ बाइल. और यहाँ पर ये जो लिपोप्रोटीन लाइपेज एंजाइम है, ये सारा प्रोसेस किसकी मदद से हुआ है? ये सारा प्रोसेस बाइल से के जरिए से हुआ है. And bile it is released with the help of cholecystokinin, and cholecystokinin is also responsible for the release of pancreatic enzyme. ये सारे pancreatic enzyme, phospholipase, lysophospholipid and free fatty acid product, and here is cholesterol and free fatty acid product. These all are digested product. Okay, now dear friends, these all digested product. And next one is mixed micelle formation. Mixed micelle formation. Bile salt, it surrounds these digested product. Bile salts, it surrounds all these digested product. Two molecules of monoacylglycerol, lysophospholipid, cholesterol and fatty acid. All these digested products are surrounded by bile salts. Okay, dear students, and here is the formation of mixed micelle. Okay, dear friends, and all these digestive products, they are absorbed by simple diffusion process. Let's see here. All these digestive products, they are absorbed by simple diffusion process. Simple diffusion process. Okay, all these digestive product absorbed by simple diffusion process. And again there is synthesis of in intestine. Again there is synthesis of triacylglycerol, phospholipid, cholesterol ester in intestine. Again there is synthesis of triacylglycerol, phospholipid and cholesterol ester. कोई रेस्टोरेंट में रिवाइज कर देता हूँ. These are digestive products. They are surrounded by bile salts and play a very important role in the formation of mixed micelle formation. And all these digestive products they are absorbed by simple different process like monosaccharides, lysophospholipid, cholesterol-free fatty acid. And in intestine, there is again reesterification process, resynthesis of triacylglycerol, phospholipid, and cholesterol ester. In intestine, there is also synthesis of another important protein, apoprotein, ApoB48. And 48 percentage concentration is synthesized due to premature termination. While there's some another important protein, they are synthesized in liver with 100% concentration. Some other proteins. Okay. All these products they are incorporated in form of chylomicron. Now let's see structure of chylomicron. Chylomicron is phospholipid, integral protein, peripheral protein, cholesterol, phospholipid, triacylglycerol, cholesterol ester. And this is the structure of chylomicron. All these digestive product, these, because there is reesterification of all these digestive product, triacylglycerol, phospholipid and cholesterol ester. Also in intestine, ApoB48 gets synthesized, only 48% concentration because of premature termination. And they all, there are some other proteins that are synthesized in liver with 100% concentration. They are also called as apoprotein. All these products they are incorporated in the form of chylomicron. It is composed of outer layer, inner layer. Outer layer is surrounded by phospholipid, cholesterol, peripheral protein, and integral protein. While there is inside there is a triacylglycerol and cholesterol ester. Okay, students. Now, with through exocytosis, chylomicron it gets transported in lymphatics and through thoracic duct it is transported into the blood. This is the absorption, and that's how the absorption process takes place. Absorption means to come in blood. Summarizing all, the digestion process occurs uh, in intestine. There's no any digestion. There's no any kind of digestion in mouth and stomach. In intestine, there is the digestion of the lipid, and in food stuff, in our food there are also there are also present triglycerol, phospholipid, cholesterol, ester. They're also present, but triglycerol is present in maximum amount. And digestion process occurs in intestine. And how digestion process occur? With the help of two important hormone, cholecystokinin and secretion. Cholecystokinin, it is responsible for the release of bile, while and also it is responsible for release of pancreatic enzyme. Secretin, it is responsible for release of bicarbonate ion, and it utilizes the acid weight of the stomach. And all digestive drug they absorb through simple diffusion, and again there is re-acidification process in intestine, and also synthesis of some protein. All these products they are incorporated in the form of chylomicron, and it is transported into the lymphatics, and through thoracic duct it is transported into the blood. And this is the digestion absorption of lipid molecule. बार बार इस वीडियो को देखें, अगर कहीं से भी समझ नहीं आती आप मुझसे कमेंट सेक्शन में पूछ सकते हैं. अपना ख्याल रखिए. थैंक यू.